Welcome to the Energy Factory. In this video we are breaking down glycolysis, the spark that fuels our body. The term glycolysis is derived from two Greek words, glykos and lysis. Glykos refers to sweet or sugar, and lysis refers to dissolution. Glycolysis takes place in all cells of the body, and it is a common step in both aerobic and anaerobic respiration. This pathway is often referred to as the Embden-Meyer-Hofparnasse pathway, or EMP pathway, in honor of the biochemists.
Gustav Embden, Otto Meyer-Hof, J.K. Parnas.
These three scientists made significant contributions to the discovery and understanding of the glycolytic pathway. Having explored the history of glycolysis, let's now unravel this fascinating pathway. Have you ever wondered where glycolysis takes place? It occurs in the cytoplasm of the cell. Glycolysis is anaerobic in nature as there is no involvement of oxygen in the pathway.
The food we eat is the major source of energy. The four main components present in our food are carbohydrates, proteins, fats, and fibers. We derive most of our energy through carbohydrate metabolism.
The EMP pathway consists of 10 steps which can be divided into two phases. The steps 1 to 5 come under investment phase where ATP is required, and the steps 6 to 10 come under payoff phase where energy is produced. To be concise, glycolysis can be defined as a sequence of reactions converting one molecule of glucose into two molecules of pyruvate. With the production of two molecules of ATP and two molecules of NADH. The step 1 of glycolysis is phosphorylation of glucose to glucose 6-phosphate catalyzed by the enzyme hexokinase. In liver cells, glucokinase, an isomer of hexokinase, catalyses this reaction. It's one of the regulatory steps in the pathway. The step 2 of glycolysis is isomerization reaction where glucose 6-phosphate containing pyranose ring is converted to fructose 6-phosphate containing furanose ring. This involves the rearrangement of carbonyl group from C1 to C2 as shown here. The step 3 is phosphorylation of fructose 6-phosphate to fructose 1,6-bisphosphate by the action of the enzyme phosphofructokinase 1. It's a key regulatory step, and the enzyme PFK1 is highly regulated by allosteric activators and inhibitors. The step 4 of glycolysis is aldol cleavage catalyzed by aldolase, which splits a 6-carbon symmetrical molecule into two 3-carbon molecules. This is a reverse process of aldol condensation reaction. The step 5 is an isomerization reaction catalyzed by the enzyme triose phosphate isomerase. The net result of this reaction is that now glucose is cleaved into two molecules of glyceraldehyde 3-phosphate. Step 4 and Step 5 together called as splitting phase. The Step 6 is dehydrogenation of glyceraldehyde 3-phosphate and its simultaneous phosphorylation catalyzed by glyceraldehyde 3-phosphate dehydrogenase. This step involves the production of NADH and a high-energy intermediate 1,3-bisphosphoglycerate. The step 7 involves substrate-level phosphorylation where ATP is synthesized from the substrate, 1,3-bisphosphoglycerate, without the involvement of electron transport chain. Here, phosphate group is transferred from 1,3-BPG to ADP, forming ATP. The step 8 of glycolysis is a mutase reaction where 3-phosphoglycerate is converted to 2-phosphoglycerate by transferring a phosphate group from C3 to C2 within the molecule. Step 9 involves the production of phosphoenol pyruvate, which contains a high-energy enol phosphate bond and very high phosphate transfer potential. Step 10, the final step of glycolysis, catalyzed by the enzyme pyruvate kinase, is the conversion of phosphoenol pyruvate to pyruvate, along with ATP production. This enzyme is highly regulated by allosteric regulators. In one cycle of glycolysis, a total of 9 ATP molecules are produced, out of which 2 ATP molecules are utilized in steps 1 and 3. Therefore, the net gain of ATP per glucose molecule in one cycle of glycolysis is 7 ATP.
Having explored the detailed glycolytic pathway, let's conclude by highlighting the crucial significance of glycolysis. During strenuous exercise, the skeletal muscles demand the occurrence of uninterrupted glycolysis. Red blood cells are solely dependent on glycolysis to meet their energy needs. These body parts derive most of their energy from glycolysis. Intermediates such as pyruvate and dihydroxyacetone phosphate are used to synthesize amino acids and fats. Reversible reactions of glycolysis are also used for gluconeogenesis pathway.
We have reached the end of our journey through glycolysis. Thank you for joining us, and stay curious.